In 2022, the Portland Harbor Community Coalition, or PHCC for short, contacted Oregon State University's Environmental Health Science Center with a request for non-target chemical analysis. This idea was inspired by some articles that were written about the non-target chemical analysis projects that were being done by the Ferguson Lab at Duke University. Fortunately, Manuel Garcia Jaramillo of the Environmental and Molecular Toxicology Department at Oregon State University has expertise in these methods. Diana Rollman, head of the Environmental Health Sciences Community Engagement Corps, was able to connect Portland Harbor Community Coalition and Manuel together for this project. In collaboration, these groups designed a study based on the questions the community wanted to explore and to suit the scientific method. Our project was funded by the Environmental Health Sciences Center and the Oregon State University Superfund Center. When Portland Harbor Community Coalition reached out, they knew that non-target chemical analysis could help us explore emerging and unmonitored contaminants in Portland Harbor.
My name is Manuel Garcia Jaramillo. Our lab specializes in non-target analysis, which allow us to identify thousands of chemicals in water samples. This is different from traditional approaches where we are just looking for a particular contaminant or a set of contaminants and using targeted approaches to look just for those compounds. With this approach, we want to have a better coverage of the chemical composition of water samples in the Portland Harbor to better inform the community about potential risks of exposure to those contaminants. The strength of this method is that allow us to simultaneously look for thousands of chemical features in the water samples. So we are not restricted for a few set of compounds, but instead we are opening our search much broader, trying to have a better coverage of the chemical composition of the water samples. Our research is aimed to improve the health of people and the environment by improving our understanding of the risk associated to the exposure of chemical contaminants, in this case in the surface water of the lower Willamette River. Community-engaged research is going to help us to break environmental injustice by providing them with the data that they need to make these changes happen.
The process for these projects usually consists of three main steps: collecting samples, processing and analyzing samples, and then processing and analyzing data. Once a month for a 6-month period beginning in June 2022, we took a boat out on the Willamette River to collect samples from 6 different sites. Our sampling sites were selected with the input of PHCC and placed in regions where the community commonly uses the river. The first site was near Willamette Park, several miles upstream from the Superfund site in South Portland. Sites 2, 3, 4, and 5 are within the Portland Harbor Superfund, and Site 6 is downstream near Kelly Point Park and the confluence with the Columbia River. We collect the water from the river using a clean bucket and bring it back to our lab in Corvallis, Oregon for the next steps. Once we have the water back at our lab, we process the water samples to concentrate the organic chemicals that might be in it. Then we analyze our samples through a process called liquid chromatography, high-resolution mass spectrometry. Basically, this lets us separate out all the chemicals in the sample and characterize them. Then we sort through the data and use softwares designed to identify the chemicals present in the sample based on their mass and charge. This data acts like a screening method. With non-target chemical analysis, we look for thousands of compounds, including those that may not be currently monitored in the river. Since this is a screening method, we're able to look for a breadth of different organic chemicals. When we look for thousands of compounds, we lose the ability to determine how much of each compound is present. There's always a give and take with chemical analysis. We look for thousands of compounds, or we can determine the concentration of a very small number of compounds. With our non-target analysis, we are able to determine what compounds are there, and we can tell if we have more, the same, or less of a compound among different sites. With this data, we can determine a large diversity of compounds and their sources, giving us an idea of where they might be coming from. Additionally, we can test our samples in toxicity models to determine if the compounds we are seeing might pose a risk to human health.
As community members who live in the area and recreate in and around the Portland Harbor Superfund site, we get a lot of questions about the river and what contaminants might be in the water, making it unsafe. We've done a lot of work to find this information, and we have advocated for new technologies to learn even more about the water. We were excited to learn about non-target chemical analysis as a way to search for chemicals that are not routinely tested for in the river. Ultimately, we want a healthy future for the river and the communities living in this watershed and beyond. This information helps us get closer to that goal.
We gratefully acknowledge the contributions and support from Portland Harbor Community Coalition, the Pacific Northwest Environmental Health Sciences Center, and the OSU Superfund Center with the development and execution of this project.